and welcome to the 10th lecture in temperature stress physiology. Today we'll be talking about chilling stress resistance. In the content of this lecture, we will be going over induction, then the chilling-induced molecular changes that may affect lipids, sugars, photostasis and pigments adjustments, proteins, and the active oxygen species scavenger systems. Under this subtitle, we will compare the enzymatic versus the non-enzymatic systems. After that, we will go over chilling injury protection and see some of the pre-harvest practices and the post-harvest practices. Chilling resistance primarily involves tolerance mechanisms since opportunities for avoidance are limited. Freezing tolerance occurs by acquisition of chilling exposure, so all cellular actions in chilling tolerance may help in freezing resistance. However, plants will take other drastic measurements to prevent freezing damage, which we will discuss in the freezing stress resistance module. Meanwhile, growers prepare their plants for chilling and freeze injuries by induction. Induction can involve several environmental changes. One way to increase tolerance to secondary water stress is by reducing watering. A common practice with transplants is to put them out of doors for short intervals to toughen them up. But reducing watering can also be effective. Mechanisms can include improvement of stomata regulation but may also involve ABA at the level of transcriptional activation. Most commonly, we think of temperature acclimation when plants increase tolerance to chilling temperatures following exposure to reduced temperatures. The rate of chilling acclimation is much slower than high temperature acclimation, frequently taking days to weeks for full expression. Acclimation can lead to a number of molecular changes, including those involving membrane lipids. Fatty acids can have increased unsaturation, contributing to a lower phase transition temperature if the double bonds are of the cis configuration. Fatty acid chain length also affects the phase transition temperature, with the melting temperature decreasing with the decreasing length of fatty acid tails. These principles are illustrated in the table from the membrane module earlier in this course. Adding double bonds to both fatty acids 18:0 to become 18:1 decreases melting temperature about 70 degrees Celsius, while shorter chains like 18 to 14 carbons lower melting temperature about 30 degrees Celsius. This relationship led to the homeoviscous hypothesis, also known as the homeophasic hypothesis. The crux being that exposure to acclimating temperatures can result in an increase in unsaturation of fatty acids in chilling-resistant plants, re-establishing fluidity at lower temperature. This acclimation change has been observed in several plants such as Arabidopsis, although some chilling-resistant plants show no change in the level of unsaturation of their membrane lipids when exposed to chilling temperatures. Another molecular change that plant cells go through is accumulation of sugars. Researchers have noticed that the levels of soluble sugars like sucrose and raffinose, as well as sugar alcohols like galactinol, increase significantly under chilling stress. This increase is caused mainly by the starch degradation pathway. Temperature stress usually leads to secondary water stress. In low temperature stress, this usually happens when ice starts forming in the apoplastic space, which leads to a decrease in the water potential there. This will cause water molecules to flow outside the cell towards the lower water potential side, leaving the cell in a dehydration state. In attempts to restore water potential inside the cell, carbohydrates are accumulated in the cytoplasm. Another benefit of accumulating sugars inside the cell is that sugars can act as active karyoprotectants for the cell membrane. Cell membranes require hydrophilic environment to keep the lipids stabilized. This is normally provided by water molecules surrounding the cell membrane. But in the case of chilling-induced dehydration, Non-reducing sugars can substitute water molecules and maintain the hydrophobicity of the lipids. Sugars can also play a role in protecting integral protein complexes. Additional aspects of photosynthetic processes can also be modified to cope with chilling in some overwintering herbaceous plants such as winter wheat, rye, and Arabidopsis. Here are some of the strategies plants adopt when exposed to chilling stress. As part of their short-term tolerance to cold, these annual cold-tolerant plants decrease the CO2 uptake capacity when temperatures drop below 15 degrees Celsius. As you remember from previous modules, plants will tend to diminish the energy imbalance by converting PS2 from a state of efficient light harvesting to a state of high thermal dissipation by taking the alternate pathway of electron transfer chain in mitochondria. Energy imbalances between light absorption and conversion to ATP and NADPH and the use of energy in metabolism and growth can be detected by a redox sensor, the plastoquinone pool. Plastoquinone is the mobile electron carrier delivering electrons from photosystem 2 to cytochrome. Short-term tolerance can recover photosynthesis by increasing thylakoid plastoquinone A content. Other strategies involve reducing the physical size and number of light harvesting complexes, which reduces the probability of light absorption. The third strategy is decreasing irradiance. This is of more than theoretical interest since plants can alter leaf and chloroplast orientation to limit interception of light energy. Finally, plants can decrease the effects of photostasis by accumulating anthocyanin pigments in the leaves and the stems of plants exposed to chilling. Those pigments are produced through the phenylpropanoid pathway. Pigments help in decreasing chlorophyll excitation by trapping light. The photo shown here is an example of anthocyanin intensification after chilling in basil leaves. Plant cells change their protein synthesis activity when exposed to chilling. Some of these changes addresses enzymes to compensate for decreased activity at low temperatures. This approach can include replacement of labile forms of enzymes with a more stable isozyme, possibly one that relies less on hydrophobic stabilization. There could also be an increase in the relative concentration of the enzyme. Since overall protein synthesis decreases at low temperature even in chilling-resistant plants, the response may involve changes in relative synthesis rates to keep metabolism from getting unbalanced. Other molecular changes during acclimation can be the production of new protective proteins. Here are some examples on protective proteins. Dihydrins, which are hydrophilic proteins that prevent membrane destabilization during osmotic contraction. Antifreeze proteins. Antifreeze proteins production is induced by chilling. They act by binding irreversibly to the surface of ice and preventing it from nucleation and forming ice coalescence. We will cover ice nucleation in the next module. As their name imply, cold shock proteins and RNA binding proteins are proteins that protect RNA secondary structure and stabilize its formation during cold temperature stress. These proteins have glycine-rich regions that act as RNA chaperones. Some of the enzymes and their products upregulated during acclimation to chilling address problems related to excess activated oxygen activity. Cells have defenses to keep activated oxygen systems in check, whether they are byproducts of normal metabolic processes or the consequence of environmental stresses. Whether they are byproducts of normal metabolic processes or the consequence of environmental stress. Antioxidant systems can function at different points. Some radical scavengers prevent damage by neutralizing activated oxygen species before they react with critical biomolecules. Other components either mitigate or repair damage. One of the strategies to combat activated oxygen species is to employ preemptive scavengers that intercept radicals and other reactive species before they react with other cellular molecules, such as proteins, membrane lipids, and DNA. Scavenger systems include enzymatic and non-enzymatic components. Superoxide dismutase, SOD, is an enzyme present in all aerobic organisms and found in almost all parts of higher plants. SOD is a family of metal-containing enzymes. It can have manganese, MnSOD, which is found in mitochondria in prokaryotes. It can have copper and zinc enzymes, which are present in cytoplasm, peroxisomes, and chloroplasts. It can have iron, such as the FeSOD, which is found in plastids and bacteria. SOD is constitutively produced but can be increased by stress. SOD removes superoxide radicals by accelerating the dismutation reaction. The second protein is catalase, which is a tetrameric heme protein that breaks down hydrogen peroxide. Catalase, or CAT, is also coded by a family of genes. Those could be CAT1 or CAT2, which are present in peroxisomes and cytoplasm, or could be CAT3, which are present in the mitochondria. Catalase undergoes two reaction cycles, which we will explain in the next slide. The first cycle is called the catalytic, which is very fast and occurs when there is abundant hydrogen peroxide. This cycle has the first one, 2 electrons reduction of hydrogen peroxide to water along with oxidation of iron. And in the second step, iron is reduced along with oxidation of a second hydrogen peroxide molecule to oxygen. The second cycle is the peroxidative cycle. This occurs when the concentration of hydrogen peroxide is low. The first step of this cycle is the same as the catalytic cycle. The difference is that iron is reduced in a two-stage process by 2 single electron transfers, shown here in step 3 and 4. This requires 2 electron donors. Peroxidases are the third class of enzymes that can also break down hydrogen peroxide. The reaction shown is for nonspecific peroxidase that transfers electrons from a variety of donors. There are also specific peroxidases such as ascorbate peroxidase or APOX, which uses ascorbate as the electron donor. Peroxidases are found throughout the cell, which is significant since chloroplasts contain no catalase. Now let's have a look at the non-enzymatic preemptive scavengers. Amphipathic molecules such as vitamin E or alpha-tocopherol can trap radicals in membranes. The diagram shows vitamin E terminating a chain reaction involving membrane lipids. Initially, the diacyl lipid is attacked by a free radical, shown here as a dotted X, forming the fatty acid radical. Addition of oxygen forms the peroxide radical. The increased polarity causes partitioning to the aqueous interface. Alpha-tocopherol is shown on the right. Alpha-tocopherol acts as an electron donor, stopping the chain reaction by forming the hydroperoxide. Tocopherol is regenerated to the reduced form by ascorbate. Carotenoids. The second non-enzymatic systems are carotenoids. Carotenoids such as beta-carotene and xanthophylls function as accessory light harvesting pigments in photosynthesis. Beta-carotene also functions in activated oxygen species defense. It scavenges singlet oxygen by by accepting the energy required to go from the singlet to triplet ground state. The energy is then dissipated as heat. Beta-carotene can also accept energy from excited chlorophyll, preventing singlets formation. The energy is also released as heat. Beta-carotene is also reported to terminate chain reactions in lipid peroxide, similarly to tocopherol. It is not surprising that xanthophylls act similarly to beta-carotene since they are formed from beta-carotene through the addition of oxygen. In the absence of stress, violaxanthin acts as an antenna pigment. Under very high light intensities or under stress, plants convert violaxanthin to zeaxanthin through the intermediate antheraxanthin. Zeaxanthin dissipates the excess photosynthetic energy as heat. In the dark, zeaxanthin is converted back to violaxanthin. One of the scavengers in the aqueous environment is glutathione, GSH. Glutathione is a tripeptide containing glutamic acid, cysteine, and glycine, with cysteine containing the thio group. The oxidized form of the glutathione is the GSSG dimer. Glutathione has several functions. It can play a role in detoxifying xenobiotics. For example, glutathione is important in sulfur transport and metabolism, being the predominant non-protein thiol. As an antioxidant, glutathione scavenges activated oxygen species in chloroplasts and in the cytoplasm. It can also act indirectly by participating in the regeneration of ascorbate and vitamin E. Ascorbate vitamin C is another water-soluble scavenger that is important in aqueous domains such as the apoplast, cytoplasm, plastids, and the vacuole. It is an important reductant for enzymatic preemptive scavengers such as APOX. Ascorbic acid usually expresses its acidity under physiological conditions, being found as ascorbate. Ascorbate can donate an electron to form the monodihydroascorbate radical. Further oxidation leads to dihydroascorbic acid. When examining ascorbate's role in battling activated oxygen species, it is apparent that enzymatic and non-enzymatic scavengers can work together in integrated cycles. Superoxide is formed in the chloroplast via the Maillard reaction when NADP, the normal electron acceptor, is limiting. SOD converts superoxide to hydrogen peroxide. Ascorbate peroxidase then reduces hydrogen peroxide using an electron from ascorbate. The reduced form of ascorbate can be regenerated in two ways. The enzyme monodihydroascorbate reductase can use reducing power from NADPH to form ascorbate, or two molecules of monodihydroascorbate can participate in a dismutation reaction that formed ascorbate and dihydroascorbate. Dihydroascorbate reductase regenerates ascorbate using reducing power from glutathione. Glutathione is regenerated using reducing power of NADPH. A bonus of the ascorbate glutathione cycle is the generation of electron acceptors in ADP that can help prevent superoxide formation at photosystem 1. As you will recall, the problem has its roots in the over-energized chloroplast. Illuminated plants exposed to stress continue generating reducing power but have less demand. This results in a buildup of NADPH and a shortage of NADP. If the electrons are passed to oxygen, superoxide is formed. A backup in electron transport can also lead to energy transfer from chlorophyll to oxygen, forming singlet oxygens. The generation of electron acceptors in ADP by the ascorbate glutathione cycle helps prevent superoxide formation at photosystem 1 as well as singlet oxygen formation. It will restore the balance between the NADP and the oxygen singlets. We will conclude the area of chilling resistance by switching from plant-based responses to cultural practices aimed to reducing chilling injury. Pre-harvest practices focus on avoidance or evasion strategies. The use of rapidly maturing cultivars allows progression from seedlings to harvest during the window between early and late season low temperatures. We can achieve a similar result for plants with longer life cycles by using transplants which spend several weeks in protected structures, effectively lengthening the growing season. Protective structures such as row covers and tunnels are used for chilling and freeze protection. The downside is the added cost of production. Delaying harvest until the crop is more mature can avoid the problem of impaired ripening following low temperature storage. This approach can also increase tolerance since many of the ripening changes, including increased membrane unsaturation, can reduce injury. Post-harvest practices have been developed to prevent chilling injury in stored fruits and vegetables. Delayed storage, sometimes referred to as curing, can reduce problems associated with subsequent chilling. Grapefruits, sweet potatoes, and peaches are sometimes held at room temperature or higher for a few days before low temperature storage. One benefit is the healing of wounds to prevent latent infections. Some fruit ripen after harvest, and the delayed storage has similar effects to delayed harvest. Temperature conditioning involves holding the samples just above the critical temperature or exposure to heat shock to allow acclimation. Temperature cycling alternates low temperature storage with short periods or at warm temperatures. Metabolic imbalances can be corrected during the warm periods, dissipating toxic compounds. Cycling may also trigger acclimation since the respiratory bursts decrease in magnitude with successive warming cycles. Controlled atmosphere literature is largely empirical, but symptoms developed frequently can be delayed by slowing metabolism and retarding oxidation by elevated carbon dioxide and reduced oxygen levels. Chemical treatments have been used as research tools to increase understanding of oxidation mechanisms, but antioxidants are routinely applied to some stored fruits to limit oxidative reactions. And since it's been a while since we watched a video within a lecture, I have found this interesting video on post-harvest techniques used for a tropical crop. I thought this would be a nice change after all the molecular information we covered. And if you like this video, you can check the Dole channel for more tropical fruit harvesting videos.
At Dole's Bananito Farm in Costa Rica, the combination of perfect growing conditions and sustainable farming techniques allows us to harvest premium bananas year-round. It takes 12 to 13 weeks from the time the banana plant's fruit stem first appears to the time the bananas are harvested. Experienced Dole bananeros measure the banana fingers on each plant to determine if the fruit is ready to harvest. Harvesting takes place 6 days a week. A team of 3 bananeros will harvest the plants in this section of the farm. Bananas are harvested while they are still green, so they can ripen to perfection just as they reach consumers. First, protective foam padding is inserted to safeguard the sensitive banana hands from spotting and other skin damage. The entire stem is cut off with a machete onto the waiting shoulders of a bananero wearing a protective pad. The leaves of the harvested mother plant are cut off, and the remaining stalk provides nutrients to the new daughter plant. The cut leaves are left on the ground where they will become nutrient-rich humus for the next generation of banana plants. The bananero carries the bunch, which weighs about 65 pounds, to the nearby cableway system. Once 75 bunches are harvested and loaded onto the cableway system, the train of banana stems is pulled back to the packing plant at the center of the farm, usually with the aid of a mule. The cableway system encompasses all the fields on our farm and is about 80 miles in total length. As the bunches arrive at the packing station, the protective pads are removed from the hands and sent back to the field to be used again. For quality control, each train of bananas is numbered and the location of the harvest as well as the harvesting team is recorded. The fresh bananas are washed with chlorinated water and thoroughly inspected. If the bananas do not meet Dole's stringent quality standards, harvesting is immediately halted in the field from which they originated. Banana hands are cut from the stem and placed in a tank of water to protect them and to remove the excess latex found naturally in bananas. The leftover banana stems and the protective plastic bags are then recycled. A gentle water current in the tank moves the banana hands to the next station, where they receive another quality inspection and are cut into clusters of 5 to 7 fingers. The clusters float to the next workstation, where they are removed from the water tank and placed on trays. Scales are programmed to accept the correct amount of bananas per shipping carton. The trays are then transported by conveyor to the next workstation, where they receive the worldwide symbol of exceptional quality, the Dole sticker. The cut stems then receive a post-harvest treatment to preserve freshness. The bananas are now ready to be packed into boxes specifically designed for long-distance transport. These boxes are assembled right on site. For quality control, packers personally identify each box they pack. Banana clusters are carefully laid in the box using methods that minimize any damage during shipment. Each box contains about 40 pounds of bananas. The bananas are wrapped in specially perforated material, which allows the fruit to ripen while in the boxes. To meet each customer's specific needs, Dole bananas are packaged several different ways: loose banana hands, single bananas, and sealed in consumer bags. A last weight check ensures that each box contains the correct amount of fruit. The entire process from harvest to box is only 2 to 3 hours. The boxes are then stacked on pallets in preparation for transport. For quality control and traceability throughout the shipping process, each pallet is given its own unique pallet identification code. As part of our commitment to sustainability, the corner boards are made from recycled materials. Sometimes cardboard is used, but usually they are composed of recycled plastic created from the bags utilized to protect our bananas in the field. Once full, the pallets are loaded into shipping containers that will take them to a Dole ship at a nearby port. As you have seen, bananas are quite delicate, so very little of our process is automated. To bring you this healthy, delicious fruit, dedicated workers provide hands-on care and attention at every stage.
So by the end of this lecture, you should know the answers to the following questions. What is induction and how is it achieved? List the molecular changes that happen after chilling injury. How do lipid changes help in chilling resistance? And what role do sugars play in chilling tolerance? How does photostasis adjust with chilling exposure? How can pigments help in chilling resistance? And what are the strategies that proteins use to protect cells from chilling? What are the main 2 subtypes of activated oxygen species, scavenger systems? And name 3 for each. Describe briefly how each antioxidant works. And finally, list some of the pre-harvest and post-harvest practices done to protect plants and fruits from chilling. Thank you, and we'll see you in the next module.